we'll move directly to the next and final talk, which is from Dame Pamela Shaw. Pamela is the professor of neurology at the University of Sheffield, director of the Sheffield Institute for Translational Neuroscience. She has held multiple positions of importance and leadership, has been one of the most productive investigators in ALS, particularly with an eye toward bridging from the clinic to therapeutics. And it's an honor to have her with us. She will talk today on subclassification and therapeutic approaches to sporadic ALS. Thank you so much, Pamela, for being with us. Perfect. So I think you're muted, Pam.
Better. Is that better? I was just saying thank you so much to Bob, Jen, and Merit for inviting me to participate in this wonderful meeting. It's a great honor to be here. So I'm going to talk about sporadic ALS and how we might begin to try and tackle that a bit better. Now, the problem is that ALS is heterogeneous, and it's heterogeneous clinically, genetically, pathologically, and prognostically. And as we heard in this morning's session, in terms of new therapy development and clinical trials, we've got to take account of this heterogeneity in the future. So some people have classical ALS as described by Charcot, but there are segmental syndromes in terms of how the patient initially presents with bulbar, upper limb, lower limb problems. There are predominantly upper lower motor neuron syndromes. And of course, some patients have concomitant frontotemporal dementia. And this audience well knows the genetic heterogeneity of the disease, more than 40 genes now known to cause or predispose to the development of ALS. And my husband's a neuropathologist. I always have to show this slide. It's also pathologically heterogeneous. So this on the left-hand side is a normal motor neuron with TDP43 largely in the nucleus, but we know it shuttles between the nucleus and cytoplasm. As the motor neurons are becoming injured, there's depletion of that nuclear TDP43 and aggregation in compact or skein-like inclusions in the cytoplasm. So 97% of ALS is that sort of pathology, but SOD1 ALS is not a TDP43 proteinopathy. They get hyaline conglomerate proteinaceous inclusions that are made up of neurofilament proteins. Fuzz is also not a TDP43 proteinopathy, and C9, the commonest genetic cause of ALS, is a TDP43 proteinopathy, but there are additional inclusions that do not stain for TDP43, particularly in the hippocampus and cerebellum. Prognostically heterogeneous, some people like Lou Gehrig have fairly rapidly progressive disease. Others like Stephen Hawking had it for more than half a century, more than 50 years. And we all know that the mechanisms by which motor neurons are injured are complex. At least 11 different damaging mechanisms happen, not only in the motor neurons themselves, but in the surrounding glial cells. And these are the FDA-approved drugs. So Riluzole, acting a bit on excitotoxicity, not a very impressive neuroprotective effect. Tofersen, a really effective treatment for the 2% of patients that have SOD1 mutations, and then Relyvrio, which is now not continued, and edaravone, where the latest evidence doesn't suggest a strong neuroprotective effect. So we've got to do something about all of these damaging mechanisms, or go right upstream and do something about the genetic upstream cause. So how do we set about improving the subclassification of sporadic ALS? I think we need to learn from oncologists who don't treat breast cancer, for example, as one disease now. They break it down into the molecular subtypes. And I think we need to do that with sporadic ALS. But the starting point, I think, is straightforward genetics. And Amar touched on this earlier. So screening every patient, for all the panel of known genes that can cause ALS pays dividends. So in the UK, we had this Ambrosia program funded by the M&D Association, 3 sites in Sheffield, Oxford, and London, and included 600 ALS patients, 300 healthy controls, and all of those. So the patients donated biosamples at the time of their diagnostic investigations and then longitudinally thereafter, and they were all systematically genotyped on a research basis. And what we found, we just looked at the first 100 patients that went into Ambrosia, of whom 7% had the expected family history of ALS. But what we actually found is that 21% had a clinically reportable pathogenic mutation, and another 21 had variants in some of the known genes that we had to say were of unconfirmed significance, so would need further work to see if they were actually predisposing to motor neuron injury. So 93% of these 100 patients had no family history of ALS, and in the current climate, 15% of those patients would have been potentially eligible for current genetic therapy trials. So I think really important that we at least offer every patient with ALS the opportunity to have a gene panel screened. Not everyone might want to take that up. And in a larger study led by Amar from the Project Mind cohort, so more than 6,000 sporadic ALS patients similarly screened, and exactly the same number, 21%, had a pathogenic mutation in one of the known genes. So we need to go beyond the family history to identify those genetic subtypes. I always like to inject a bit of hope into my talks, and I think I want to show just 2 videos from effective therapies for motor neuron degeneration. And I think we can take hope from these genetic therapy trials that maybe we'll also get there with sporadic ALS patients. So the first one is the tofersen phase 1, 2, and 3 trials that we've heard a bit about earlier today. And I'm just going to show you the first UK patient that went into the tofersen trial. He's been having monthly tofersen intrathecally for 8 years now. When he first came, he was in a wheelchair, could just about bear weight with 2 elbow crutches, but couldn't really walk. And then this is him. So during the trial, he could walk up his garden steps for the first time in 2 years. And in the open label extension, which is walking around his house with no walking aids whatsoever. So dramatically life-changing therapy. Many of the relatives in his family had died within 2 years, and he's still going strong 8 years on. The second one I want to just show you is a different type of genetic disorder. So the autosomal recessive problem of spinal muscular atrophy, a childhood form of motor neuron degeneration, which is caused by a deficiency of the survival motor neuron protein. And preclinical studies, including from our center in Sheffield, showed that if you can use gene therapy to replace that SMN protein, you have a dramatic effect in the mouse, giving confidence that that might work in human beings. And this is a lovely story of a family in the US who had a little daughter with type 1 SMA, floppy baby, Verdnick-Hoffman syndrome. You can see what a bright, intelligent little girl she is, but she died at the age of 15 months. The parents had a second child, really hoping she would be fine, but she also had type 1 SMA, Evelyn, but she had the chance to have Zolgensma treatment at Ohio State University, the Nationwide Children's Hospital, and this is what happened to Evelyn. Sitting up at 2 months, rolling over at 3 to 4 months after treatment. Cheeky little girl. And then at the age of 3, running about like a normal toddler, actually dancing with her neurologist, Jerry Mendel. So I'm showing these really that if we get the right treatment, motor neuron degenerative disorders are treatable. And I think we should take heart from that for sporadic ALS. There she is, very proud of herself. So I think we've learned a lot from recent trials, as we heard from Sheena earlier. And one of the really important things to come out of the tofersen trials was this lowering of neurofilament light in both CSF and plasma. So if we've got a treatment that's working, and the clinical indices showed that tofersen was working, we're protecting the motor neurons and causing less release of those neurofilaments into the blood and CSF. So can we use that insight to do better in terms of sporadic ALS? And as we heard very elegantly described earlier in this meeting, we can now, in the last few years, for the first time, model sporadic ALS. So you can make iPS-derived motor neurons by taking skin cells or PBMCs. We also use a different direct reprogramming method without going to the embryonic stages of development. And there are pros and cons of both approaches, but the advantage of the direct reprogramming method through induced neural progenitor cells is that those cells that you generate retain the epigenetic features of aging and they're non-clonal. So the tofersen trial suggests that neurofilament light might be a good biomarker of therapeutic efficacy. And can we use that, as we discussed earlier this morning, to do shorter trials, but also the possibility of picking out responder subgroups from a heterogeneous trial population? We may have thrown out effective treatments in the past because we couldn't pick out responder subgroups clinically, but can we do it based on those that get a lowering of neurofilament levels? If you take Herceptin for breast cancer, the overall trials in a heterogeneous breast cancer population, it didn't work. If you direct it to the right molecular subtype, it does work very well. And I think neurofilaments also are allowing us to stratify patients at baseline based on disease progression rate. So the higher the level of baseline neurofilaments, the faster your disease progression is. We can dig in to that population of fast and slow progressors better now to understand what the genetic determinants are of that. And this is confirmed in a recent trial, the MIRACLES trial of low-dose interleukin-2. So in this case, it was phosphorylated neurofilament heavy rather than NFL, but the higher the level of that neurofilament protein at baseline, the faster the disease progression rate. And that, when we see the full published results of this trial, made a difference to treatment response. So what about sporadic ALS itself? So, and we reviewed all the pipeline of clinical trials and preclinical things in development just earlier this year. This is the pipeline of genetic therapy trials. SOD1, really a trailblazer, I think. C9orf, we need to go back to the drawing board. Fuzz also looks very promising, but some of these genetic therapy trials have attempted to tackle sporadic ALS. So the lowering of ataxin-2, as we heard from the Biogen presentation, that didn't work at the 6-month time point, so that program has been terminated. But the Curalis trial, which is using an ASO to try and restore stathmin-2 levels, is ongoing. So some genetic therapy approaches might be helpful for sporadic ALS. And just briefly, in terms of the pipeline preclinically, all these mechanisms I've targeted in red would apply to sporadic ALS. So increasing neurotrophin levels, decreasing excitotoxicity, addressing dysregulated RNA would all apply to sporadic disease. We reviewed also the pipeline of small molecule therapies, and you can see, I think we saw a graph of the number of trials, 25 trials in the US now for ALS, and I think it's a real source of hope that so many companies and so many academic labs are actually developing new therapeutic approaches. You can see from the mechanisms of action of these small molecules, there's a great interest in neuroinflammation and also proteostasis. I'm going to tell you in the last few minutes about a pathway, a therapeutic pathway that we pulled out from our research institute, Citran, and you'll hear more about this from Eclipse Therapeutics in the industry day tomorrow. So very early on, when we were modeling ALS, putting SOD1 mutations into motor neuron cell lines and so on, we found a dysregulation of this NRF2 pathway. Not only NRF2 itself, but all its downstream genes were downregulated in the presence of ALS causing mutations. And that's a stress response system. It should be upregulated when a cell is under stress. And you could see the same in CNS tissue from ALS patients. Genes and proteins controlled by NRF2 were downregulated in ALS. And what NRF2 normally does, so it's a transcription factor held in the cytoplasm by this other protein called KEAP1. When the cell is under stress, KEAP1 releases NRF2, it gets into the nucleus and upregulates a whole host of cytoprotective genes. And that system is not working properly in ALS. And there's a whole host of evidence, which I won't go into. Particularly interesting was the study done on two different strains of SOD1 transgenic mice, one with a slow disease progression, one with a very fast disease progression, with exactly the same burden of mutant SOD1. And the slow progressing mice had a good upregulation of that protective system. The fast progressors didn't. So if we could, so at the moment we have riluzole acting on excitotoxicity. If we could upregulate that NRF2 system and its downstream genes, we would protect motor neurons from all the points of damage shown by these green arrows. This is looking at it in another way. If you look at all the damaging mechanisms, the key molecule that we pulled out that activates NRF2. It's S-apomorphine, now known as M102. It activates this NRF2 pathway, but we also found, as you'll hear tomorrow, that it activates another transcription factor, heat shock factor 1. And together, those two pathways protect against a lot of the damaging mechanisms that are happening compared to riluzole, edaravone, the Amylyx drug that act on one or at the most two of the pathways. So we pulled out from a drug screening program this NRF2 activator. And we partnered with Eclipse Therapeutics in the US, and together we took that from academic studies with applying together for funding, and we got funding from the Medical Research Council in the UK, FightMND in Australia, and the Department of Defense in the US. So we needed to raise £5 million. We showed, and I won't show you all the detail, you'll hear more about this tomorrow. We showed that this compound improved motor neuron survival under stress conditions in vitro. It improved the motor function in two different ALS mouse models, SOD1 G93A and a TDP43 mouse model. We showed evidence that it gets across the blood-brain barrier and activates genes controlled by those two transcription factors. And we got orphan drug designation. We needed £5 million to do the GMP manufacture and complete 1 month toxicology. But we've done all of that now. I'll just show you some brief highlights of how this drug works. So I mentioned before that we quite like to do direct reprogramming of fibroblasts to make astrocytes and motor neurons. And we've particularly looked at making astrocytes from ALS patients and growing healthy motor neurons lit up with GFP on top of that astrocyte layer. The ALS astrocytes are toxic to the co-cultured motor neurons compared to healthy controls in blue, and that's C9 in green, sporadic ALS in pink, SOD1 in purple. So we've used that astrocyte toxicity as a screening method. And in terms of rescuing motor neuron survival, this M102 molecule does really well in the different subtypes of ALS, and it outperforms riluzole, edaravone, and monomethyl fumarate, which is the active compound from dimethyl fumarate, used as an NRF2 activator in multiple sclerosis. So it protects motor neuron survival. It also gets rid of TDP43 proteinopathy. So in the astrocytes that I showed you, if they're from sporadic or C9 ALS, we get this abnormal fragmented 35 kilodalton band, which is not there in healthy controls. And if we expose those cell cultures to M102 for 48 hours, we get rid of that abnormal band and restore the level of full-length TDP43, and that's shown in a spaghetti plot on the right-hand side. We also used RNA-seq in those astrocytes to see what pathways are altered by M102, and these are the top pathways: oxidative reduction process, VEGF, inflammatory response, autophagic response, cell adhesion. And again, those positive gene expression changes are there in the different subtypes. We could look at the heat maps from the astrocyte transcriptome, and we could pick out good and less good responders. So in this heat map on the right, you can see healthy controls. Next to it is the transcriptome of those that got a really good response in terms of motor neuron survival. And then how in those high responders, the heat map of the transcriptome normalizes itself to almost become the same as healthy controls, whereas the low responders don't get such a good response. So we're ready now to go first in human trials. We'll go into healthy volunteers first and follow that rapidly, we hope, with a phase 2 trial in ALS patients. So to end, the upstream genetic cause of ALS can be identified in at least 21% of patients, including some patients who appear to have sporadic disease. New clinical trial approaches need to take account of the heterogeneity of ALS and embed experimental medicine biomarker components. And we need to keep on chipping away at the molecular subclassification of sporadic ALS. We're beginning to find biomarkers of therapeutic efficacy. Neurofilament levels are the leading candidate, of course, but can we use those biomarkers to find responder subgroups amongst the sporadic ALS patients? Some genetic therapy approaches that have worked so successfully in SOD1 and SMA are currently being applied for mechanisms operating in sporadic ALS. And small molecule therapies, I think we need to definitely build on riluzole. So we either need a cocktail of drugs that tackle multiple mechanisms or a single drug that will correct multiple damaging mechanisms. And I've shown you the brief example of M102 that you'll hear more about tomorrow. So I just want to end with the exciting concept that motor neuron degenerative diseases are treatable, as exemplified by SMA and SOD1 ALS. And this gives hope, I think, for our ability to find more effective therapies for a broader group of ALS patients. So I'm going to end there now. Just thank you again for asking me to participate, and thank you to all the funders that support the work and the great teams within Citran, where we collaborate together to try and harness exciting developments in neuroscience, but translate those into benefits for our patients across the road in the Royal Hallamshire Hospital in Sheffield. Thank you.
Thank you so much, Pamela. That was really wonderful. I think, as if we didn't need motivation enough, those very moving videos are unforgettable. It's as simple as that. And thank you for those. But more importantly, I think that transition you've made, bridging with all of your clinical work in Citran into an actual therapy now entering trial is exemplary and extraordinary. So thank you for showing us these data. We look forward to the presentations tomorrow as well. So we might now have questions targeted to you specifically, and then perhaps some general questions based on the whole day. We have about 45 minutes or so as needed to discuss this. I think one of the questions that arises, which is in the chat, is, are factors known that are responsible for the low NRF2?
It's certainly not fully understood yet. There is evidence that high levels of oxidative stress can impair that system. There are, Bob Bowser found a molecule that binds to KIAP1 and prevents the system from being activated properly. So we haven't unraveled it completely, but it's definitely there across all the different subtypes, that somehow that stress response system is not operating properly. And we know that that system declines with age as well. So your NRF2 system becomes less efficient with age. It's one of the effects of aging, just like our mitochondria aren't quite so efficient. So it may well be one of the factors that kicks in as we age over the decades.
And if I may ask, at how many levels does the S-apomorphine interact? I mean, does it really have one primary major mechanism of action, or does it affect, is it pleiotropic? Does it affect multiple aspects of the cell?
So as far what we know in terms of so NRF2 is normally held by that KEAP1 protein, and when the cell is under oxidative stress, the cysteine residues of KEAP1 get oxidized, and normally KEAP1 is feeding NRF2 to be repeatedly degraded all the time. So it links it to the ubiquitin proteasome system, so it's continuously degraded. When KEAP1's cysteine residues get oxidized by an electrophilic compound like M102, then that stops happening. So the newly synthesized NRF2 it gets upregulated and is able to get into the nucleus and upregulate all those genes. There are, apart from KEAP1, the regulation of NRF2 is quite complicated. Lots of things contribute to it. So in the nucleus, there's a protein called BAC1 that can inhibit NRF2 acting on the antioxidant response elements of the genes that it targets. So it's a complex regulatory system, but we think that M102 primarily is acting by altering KEAP1 so that NRF2 can get into the nucleus. And we've got a paper in press that shows all of that. Does that answer your question, Bob?
It really does. Yes,
So,
it really.
yeah.
I mean, the other part of the question is, are there sort of off-target effects? But clearly you have also very strong animal data already suggesting that M102.
So we've done 1 month, so far 1 month, and the 3-month toxicology are going to happen soon. But we've got 1 month toxicology in both rats and non-human primates, and it's very safe, well tolerated. If you use a massive dose, 18 times higher than what you would be predicting to use in human beings, you can get a bit of liver dysfunction. And omaveloxolone, which you may have come across, is an NRF2 activator that's just been licensed for Friedreich's ataxia. And that is a powerful NRF2 activator, and it causes liver toxicity in 37% of patients. So raised liver enzymes. But M102 doesn't do that. It doesn't affect the liver unless you use it at a very high dose. So it seems to be very safe. The extensive non-human primate studies showed that it looks fine.
Thank you. A question from chat. Luca Gianni asks if NRF2 activation has any effect on TDP43 splicing events.
So that's a very good question, and we're just looking at that. So we've shown it gets rid of the small molecular weight, the 35 kilodalton fragments of TDP43, and we're looking now, so we haven't got the data yet, but we're looking to see if it alters the splicing dysfunction that happens to UNC13A, STATHMIN2, etc.
Interesting. So may I ask a question about your culture system? The astrocyte bed on which you plant the motor neurons, those are normal astrocytes?
So we take them from healthy controls and then from the different genetic subtypes of ALS. So what I showed you were cultures from C9 patients, SOD1 patients, and sporadic ALS. And as a primary screen, on top of the human astrocyte layer, we put healthy mouse motor neurons that came from Tom Jessel's lab that are lit up with GFP. Yeah. And we can measure the motor neuron survival over 3 days. And we get a dramatic difference between the different ALS astrocytes compared to the healthy controls.
So the probe you're using is pivotal in terms of astrocyte function in this.
It is, and the NRF2 system is known to be much more active in astrocytes compared to motor neurons. So motor neurons don't upregulate it very well. They seem to rely on the NRF2 system activating in neighboring astrocytes, which, for example, then causes the astrocytes to release glutathione and other factors that preserve motor neuron health.
Thank you. If
Yeah.
I may, just one other closely related question is, you demonstrated 5 pathways which appear to be benefited by the M102 therapy,
Yeah.
beginning with neuroinflammation. But those were assayed in astrocytes. My question is, what about the profile in the motor neurons on top of those astrocytes?
So yes, we haven't done it. So the primary screen is with the healthy mouse motor neurons. We can then have human on human motor neuron astrocyte co-cultures. And we have we've made directly reprogrammed eye neurons, exposed and non-exposed to M102, and we have the RNA-seq data, and we're just analyzing it. So I can't show you that yet. We've got the transcriptome data in astrocytes. Yeah.
Thank you. So if I may, I'd like to ask a general question to all of the speakers, and indeed people who may not be speakers, and that is this question about discerning an abnormal phenotype in IMNs, eye motor neurons derived from sporadic ALS. So there seems to be an emerging consensus, and it was just brilliantly presented by Justin Achita, of course, that if one looks at the right parameters, sporadic ALS motor neurons in culture are not normal. And do we accept that? Has that been looked at in a blinded manner? Is that clearly the case? Because clearly that's, and Justin spoke about this, that the implications of that are phenomenal in terms of suggesting that this type of cell derived from a dedifferentiated precursor cell, presumably stripped of most epigenetic factors and then redifferentiated, still has something heritable that reflects the properties of the donor. So how do folks feel about that?
So Justin may be the best person to answer that. From what I think some people who work with iPS-derived motor neurons struggle to see phenotypes. But Justin very beautifully showed that there are phenotypes that you can detect. I was going to ask him how old the cultures were. How long did he have to keep them going to be able to see that those phenotypic changes? Maybe Justin could.
Yeah,
please.
I think they definitely become more prominent in the 4 to 6 week time point of culture. So I think if you're able to get them out that long, that's where you'll most reliably see the differences. And one thing that helps with that is to just keep them in a 3D spheroid. In that way, you don't have to worry about them lifting off of the Petri dish.
Yeah. And you can certainly see electrophysiological abnormalities in those iPS-derived motor neurons.
Yeah, we haven't looked at that for some time, actually. But yeah, in the initial days, there was some hyperexcitability of the ALS lines. Yeah, that we did observe. Brian Wanger was somebody we worked with at MGH to look at that, actually. Yeah.
But I worry that, you know, the question is how reproducible are these findings? In other words, I think what you've published, Justin, is incredibly impressive, and it sure looks as solid as anything ever is. But my query is, I mean, I guess this is the really best way to phrase it. It may be that very particular approaches have to be taken to find that phenotype. And you have identified those and used them brilliantly. Maybe those people have trouble finding the phenotype in a sporadic IM and simply don't quite know how to look for it. But I worry about the variability across the field in that observation. I don't know.
Yeah, it's an important point and sort of big issue. There is actually an initiative that's just getting started. It's actually started by an ALS patient, but it's called Project Mosaic. And the goal is to get a couple of these protocols for iPSC motor neurons where people have seen, reproducibly seen ALS, sporadic ALS lines having phenotypes as well as the familial ones, and to get it kind of set up at, you know, CROs who can then allow anybody to use them to test compounds and hypotheses. And so I think we're starting with the one that I showed today, and also one by Alyssa Coyne and Jeff Rothstein. So, you know, if this all goes well, then we might have a couple of these models at least where, you know, anybody can foreseeably use that. Yeah.
I wonder if I could ask any of you, perhaps Eva would have thought about this a lot also. To what degree are the iPSCs really stripped of epigenetic marks from the donor? I mean, it's sort of glib to say, well, they're stripped of epigenetic, and therefore there must be something in the genome. But in fact, are they really stripped? Are they really sort of de novo unmarked cell types?
I could address that, and that we have not yet looked, but that, and I don't know if others have, but our impression is because we see a lot of variants. We've created, I think, you know, in our own lab, about 18 sporadic iPS cell lines. And they clearly all have epigenetic modifications, but what we need to do is understand how efficient our quote stripping of these epigenetic modifications are. I would love to know other people's input, because we actually feel that there is a lot of epigenetic modifications of these lines.
Yeah.
But we don't use them experimentally as much as other groups do. We're just starting to do that.
On that topic, I wanted to ask you in particular whether you've looked at the toxins you've implicated in these pretty remarkable epidemiologic studies essentially as cell type specific toxins for cells in culture.
And no, we haven't. That's one of the reasons we think maybe we should do it. We don't, you know, I mean, what I'm most commonly asked for is, have we started to do this work in mice? And we actually proposed it, and we ran into so many issues with our animal group in terms of getting approval to, you know, to getting our institutional animal commission to approve some of these proposed studies, that we've taken a step back and are asking or wondering about specific cell lines. But no, none of that work. We have not done it. And to my knowledge, it has not been done.
And I know the Parkinson's field are using zebrafish in that way, kind of exposing zebrafish models to various pesticides and environmental toxins.
Yeah, and you may know, Pam, but we published a long time ago, we made a zebrafish model of ALS, an SOD1 model, and we published that, excuse me, and we published that, but the animals didn't really thrive. So I kind of abandoned that too. It sounds like I abandoned a lot of things. It's just when things really aren't working well, I kind of move on. But I'm hopeful that the cell lines, I'm hopeful that the cell lines might be more beneficial.
Yeah,
it's a great question.
And with respect to cell type specific effects, I wanted to ask you, Aaron, you did the work long ago in which you studied a conditional either knockout or inhibition of dicer, as I recall, and that was in motor neurons. Is that correct?
Yes, thank you, Bob, for the question. So, yeah, we did a dicer conditional allele crossed on a motor neuron specific area. I briefly mentioned it also in the talk.
Yep.
And yeah, so I other than the fact that we did it and it phenocopies many facets of ALS, do you want me to elaborate on anything specific?
I'm just curious as to whether suppression of dicer in other cell types would have some clinical manifestation.
Right. So dicer is such a prominent and important, you know, a key machinery that it dramatically affects almost any cell whereby it is knocked out or knocked down, reflecting on the importance of microRNAs primarily. And then I think this might also reflect on a high evolutionary conservation and selection against variants. It's probably is embryonic lethal. At the same time, where people crossed the dicer allele to different regions of the brain, to create deletions in different regions of the brain, very often the consequence is in neuronal death, namely neurodegeneration, although it's not always the case. There are some places where neurons are, keep living on apparently without microRNAs after they have been differentiated. Yeah, I think, at least for me, the interpretation of a dicer experiment is in the context of what we see in humans, in the context of the global downregulation of dicer and microRNAs we see in human, and then the interpretation makes sense. Then we can say we can directly show the necessity of dicer and microRNAs for neuronal survival.
Thank you. Yeah, I also, again, thinking about these uses of these incredible iPSC-derived motor neurons, I wanted to ask you, Dr. Zhu, whether or not you had undertaken any studies of somatic mosaicism in cultured motor neurons as compared to whole brain. And
No,
does
we
that
haven't.
change over time?
No, we haven't done that, but I think it's well known that some iPSC lines have like a cancer mutation. So that's a major reason that we didn't want to use a iPSC model to study somatic mutation, because we worried that the cancer mutation may cause genome instability.
Yeah. So, to me, one of the basic questions is, why is survival decreased in these sporadic iPSC-derived motor neurons? And why are we getting increased somatic mutations in general in ALS? What does this tell us about genome stability as a primary deficit that could underlie both familial and sporadic ALS? And how do we test it? I don't know if, Amar, you've considered those issues.
Yeah, so genome instability is obviously very important. And somatic mutation, I mean, I loved the talk on somatic mutation. I thought it was fantastic. But it's, as you saw in the talk, it's actually very difficult to identify at any level, because if you go down to single cells, then the probability that one has a relevant somatic mutation is low. And if you look at large clumps of them, then it's diluted by all the non-mutated ones. So I think it's very challenging. One of the things I thought was interesting, sorry to slightly diverge from your question, one of the things I thought was interesting in that talk was the idea of the prion spread, because there are other ways that it could spread. And neurons, for example, are phagocytic. So it's possible that when a neuron dies, when that if there's genomic instability and the first neuron just becomes debris, then as that's phagocytosed by nearby neurons, that goes up into the cell body, and that's the spreading mechanism from neuron to neuron by phagocytosis. That was shown, I think, by Linda Greensmith or Joe Martin at UCL a few years ago, that they're phagocytic. I just wondered whether that is something that you've seen.
Yeah, I leave that open for anyone else on the conference or anyone else in the panel. I guess, though, one of the questions in my mind hearing these talks is, how can we quantify genome instability? I mean, oncologists have been thinking about this obviously for 40 years. This is their bread and butter. And what does it mean in the context of neurodegeneration as opposed to, say, cancer? What are the hallmarks? What are the triggers? And ultimately, of course, if that's an underlying theme, how can we interfere with its biology to correct the disease? And I guess there are all kinds of measures. One can look at mutation rates, for example, in cells in culture. One can look at DNA damage. There are all kinds of assays to look for DNA damage, as others probably know better than I do. But I wonder if it's a corner of ALS that deserves further investigation, because something these are not what I'm taking. Again, is that these sporadic motor neurons are not normal. There is something that reflects their donor that has somehow been heritable, that also tells us something about pathology. And I think it's as fundamental as deficits, again, in something about genome organization and stability. I had thought, maybe I misunderstood you, Dr. Zhu, that you were able to do analysis of single neurons that you cell sorted. So in fact, that was a way to address that problem, Amar, I think, of looking at mutational issues in single cells. Is that right, Dr. Zhu? You were able to do that, yeah.
Yes, the previous mutation we did was single sorted neurons. So I was also thinking about how to address like this genome instability issue in neurons. So I think one future opportunity is using the third generation of sequencing, which is the long-read sequencing, especially the nanopore sequencing, because people may know that nanopore sequencing can also tell you, based on the model of the sequencing, the charges of the sequencing, and also the time lapse of the bases, you can actually tell whether there is a modification of the DNA. It can be like a DNA damage or it's epigenetic modifications of the DNA. So, and also the long-read sequencing is can be based on a single DNA molecule. So you actually know that whether it's a private event.
Yeah, excellent points. So what I'd like to do in the last few minutes is literally ask each of the speakers, where do we go now in terms of understanding sporadic ALS? And there'll be some common themes, but I suspect there will be some individual flavor that will suggest different directions. And just taking speakers in their order in this day, let me start with you, Amar. So you've got $10 billion given to you by someone, and it's yours to use in any way you want, only if it will help us understand sporadic ALS. Where do we go?
So I think the very first thing, the thing that's really been missing, which lots of us are trying to address, I think we're making some good progress in the UK, is proper linked data at multiple levels from each person. So at the moment, we've just got, for example, population registers, and then you have to go out and see, can you find the environmental exposure of that person? Can you sequence them? What we really want is absolutely deep and total phenotyping of every single person and complete capture in the population of each person with their full history, linked medical records, etc. If you have that depth of information on thousands of people with ALS, and ideally do that internationally, then I think it would become a much more tractable problem, because you'd understand where do you have to go from here, you'd understand the clustering of the subtypes, you'd understand what do these iPSC neurons reflect, is it something that they've been exposed to, etc. What's the genetic burden, all of those things. So I think really ultra deep phenotyping of huge cohorts of people by linked data.
And presumably longitudinally?
Absolutely, yeah, longitudinally, definitely.
Yeah. Thank you. Aaron, sir.
Yeah, thank you. So first of all, I think Amar gave an outstanding answer, and I completely agree. I would just add a few nuances. So for the idea that you need linked data, so you need a variety of data sets. It talks to generating a multi-omics layers of information, to name a few, proteomics, metabolomics, transcriptomics, epigenetics data, etc. And of course, the genome, right? That's I would say the basic one. And then you need data science technologies that allow you to do the links. And these rely on very often on the ability to handle data in a way that's classic statistics, become a limitation. So there are means to handle big data sets in a way that would also allow to finding the connections between the features, some of which might not necessarily be linear. So maybe the buzzword here would be AI or machine learning. As for IPCs, I just want to mention something that is related to a project we developed over the last few years. I did not talk about it at all, but it's something that is actively going in my lab, and that's allowing a very deep phenotyping of iPS-derived neurons by advanced microscopy, by having very many multiple channels of information from microscopy. I think when this would come into play along with other omics, and we would have an effective way to take all these sets of data together, I think it would be very valuable. So in a sense, what Amar has said.
But at an remarkable in vitro level. Yeah.
Yeah, I agree.
Thank you. That's terrific. Justin.
Yeah, it's a great question. To me, what I would love to be able to utilize is more patient data linking genetics to progression rate. I think this is probably included in what Amar was thinking, but I think particularly for me, the progression rate data is challenging to acquire, but also really valuable in identifying good targets. It could be maybe neurofilament levels might be a good one to do. Might be better than survival itself or easier. But something to think about there. And then I would like to see some biomarkers that are more or that change more rapidly potentially in the clinic. So neurofilament I think is a great one. But as in the tofersen trials, the change is over maybe 6 months. And so could things like the TDP-43 cryptic peptides or, even a TDP-43 seeding assay from the CSF of patients get more like a more sensitive and potentially faster change signal in the clinic that could tell us if we're going in the right direction. And then I would say an extension of that is I think the Healy folks are already thinking about this, but maybe if we could extend the modalities to something like oligonucleotides, which I think have the potential to really quickly assess new targets for their efficacy in human patients, that would also be tremendous. So those are kind of the things that I would love to see.
Thank you. Thank you. Eva, please.
Yes, 3 things. One, I echo what Amar said and would love to see that. Secondly, I'd like to, again, get on my prevention hat and tell you that, well, I was able to get funding for 4,000 patients, 4,000 people to enter them in a prospective trial, people who are at a very high risk of developing ALS based on their occupation. It would be really nice to make that 40,000, wouldn't it? And really have a large population of individuals from more diverse backgrounds, multiple occupations to follow prospectively to really address many of the questions that Amar has raised. And third, I would love to have enough funding to internally, because our patients are so well phenotyped. We see them every 3 months. We have their progression rates. We have, sufficient funding. We've done epigenetics. I would love to do transcriptomics and ATAC-seq. It would be really wonderful. We've done metabolomics at 3 time points. It would be wonderful to have the funding to more deeply dive into this well-characterized patient population that we have and continue to grow it. So really funding, Bob, as you said, becomes limited, even more so in our case than capturing the patient population and the deep phenotyping.
Thank you. Thank you. Fantastic. Let me ask you, Dr. Zhu, your thoughts. How do we finally understand sporadic ALS, and how important will somatic mutagenesis be in that?
Sorry, how important the somatic mutations in sporadic ALS?
How big a part will the concept of somatic mutagenesis play in our understanding of sporadic ALS?
So based on our current result, I think it's only a small fraction of the sporadic ALS cases. But I think, as I mentioned in the talk, I think it's definitely limited by our sequencing technology. So I think with the advances in the technologies, we would be able to identify more and more somatic mutations in the sporadic cases. And this is basically based on the prion theories of the TDP-43 aggregates. I think I don't know if anyone has tested how many what is the burden of TDP-43 aggregates you need to initiate this the event. I think there are perhaps some like animal studies suggest that you can actually super dilute this TDP-43 aggregates and still have this like a progression of the spread. So if that's the case in human, then you probably only need a few cells that actually cause this initial lesion, right? And detecting a somatic mutation in these few cells is going to be impossible at this moment. I think maybe in the future we can do that.
Yeah. Let me ask you, apropos of your own work, you focused mainly on exome analyses, exome itself. What do you think about generalizing the search for variants that are somatically derived across the full genome?
Well, I do think it's going to be useful. The reason that we only focus on the exomes is because they are easy to interpret. I think with the AI helping us interpreting the mutations in the future, I think sequencing the whole genome will be also useful.
Yeah. Thank you. That's terrific. Pamela, please.
I think
Take us home.
I think
Take
all
us home.
wise things have been said, largely. So I totally agree with Amar that having in-depth data, longitudinally gathered, deeply phenotyped, genotyped patient cohorts, that, one thing that really interests me is in the genetic subtypes. How do people keep that damaging gene at bay for 50 or 60 years? What are the compensatory responses to the stress of the presence of that gene that keep the person healthy for decades, and then disease develops? The trouble is we can't do that in sporadic ALS. We can't identify them pre-symptomatically, although the UK Biobank has 600 ALS patients now, most of whom signed up and gave their samples and their data when they were healthy. So, that represents an opportunity to kind of see what's happening before the disease onset in patients with sporadic disease. I agree totally with Justin that multi-omics, so I showed you astrocyte toxicity. We've actually reprogrammed astrocytes from a big group of sporadic ALS patients, and they vary in some astrocytes from ALS patients are as good as healthy controls. Some are severely toxic, and there's a range of toxicity. So we're digging in with RNA-seq and omics techniques to see what the differences are. And there's some very interesting differences, including the NRF2 pathway is much more impaired in the severely toxic astrocytes. So, and I think one thing we can get a handle on better now with the neurofilament data is fast and slow disease progression. So, profiling patients to understand what the genetic factors are, the gene expression factors that determine fast versus slow disease progression. One thing that worries me, though, what if sporadic ALS requires subtle changes in about 5 genes?
Oh, exactly.
And you're not going to pass all 5 of those variants to any one of your children, so it doesn't appear familial, but it's still genetic. How do we? And yet, if you have only 1 or 2 of those, you're perfectly healthy. So how do we get at the fact that there's probably multiple subtle genetic factors that make your motor neurons more vulnerable?
So I was going to ask Amar to address that, because it has been said until recently, correct me if I'm wrong, that we do not have the computing power on the face of this earth yet to do full studies of combinatorial genetics, but we have to at some level, because that may be the bridge out of Mendelian genetics. And Amar, can you help us understand where we are? Because Pamela, I think, is so right that we have not really tackled that problem in depth. We saw Dr. Zhu show the fact that some of his C9 cases had multiple mutations, as did the SODs, and we've all seen that.
Yeah.
But how can we look at that systematically?
So there are two aspects to what Pam has said. So the first is, if you've got 5 genes, if it's the same 5 genes that always have to have those variants, that's a much more tractable problem. If it's a particular subset of say 30 genes and any 5 of them would do it or something like that, then it's more difficult. But actually, it's still not impossible, because it depends on what you mean. If you're talking about an additive effect, so each variant simply adds to your risk, and then when you reach a particular threshold of liability, you get disease. You can find that with a polygenic risk score, a bit like Eva showed. So that would be a fairly straightforward thing to do. But if you're looking for an interactive effect, what we call epistasis, so that you have to have, it's a bit like a kind of dominance recessive relationship across genes instead of at the same locus. That's a much more difficult thing to do. And you're right, Bob, the possible number of combinations across 3 billion base pairs is ridiculous, even if you're just doing pairwise epistasis. But there are ways you can reduce the problem statistically, and people have tried to do that. So there's a group in the UK called, not Precision ALS, but Precision Life, that's what they're called. And they have a particular bespoke, we're not allowed to know what the method is, statistical method where they look for these combinations of genetic variants, and they claim to find clusters of genetic variants, and they've got good evidence from other diseases that what they find makes sense, and therefore their statistical technique seems. And they've now applied this to ALS. We haven't seen the results yet, but I think they'll be presented in Montreal.
Wow. Okay. Thank you.
And there's a company called Genius, isn't there, in Australia, that's attempting that sort of thing as well.
Wow.
Yeah.
That's interesting. Well, like you, Pamela, and I suppose most of us on the call, although we have, all of us worked side by side for 40 years to only see 3 drugs that work in ALS, 1 approved and then not approved, I still remain very, very optimistic. And I feel that way, both because the depth and breadth of the ways we're trying to struggle, particularly with sporadic ALS, have never been richer. The technology's never been better. And most importantly, or as importantly, we are in an era of incredibly innovative new therapeutics. And I know a lot of that's nucleic acid based, but some of it isn't, as you've shown us. Some of the more traditional approaches can yield very, very exciting compounds. So I at least remain extremely upbeat. So look, I think from my perspective, go ahead, please. Please.
Me too. No, I was just saying me too. I think glass half full.
Exactly. So from my perspective, that pretty much draws this session to a close. I'd like to turn things over now to Jen for any comments she may have at the end of day 1.
Thank you. My mind is so boggled and full of such hope, but it's all such interesting topics and things that many people just I don't think are even aware that a lot of this stuff is being worked on. So that's why, conferences and symposiums like this are so important. So I can't thank you all enough. This has been really enlightening. And I do want to tell everyone that each of our sessions have been recorded today. So they will all be parsed out and put on our website and sent out to all of the registered attendees. If anybody is for all of the ones that we have approval to share, we will, and that goes for the next 2 days as well. So speaking of the next 2 days, join us tomorrow beginning at 9 a.m. Eastern Time. We'll have presentations from 19 pharmaceutical companies about current research in ALS that they have underway. We will actually start the day with an announcement of the 2024 Lalji Family Student Scholar Award and hear about the work that earned that special award. So that'll be a special treat in the morning. It'll be a really engaging and packed day. And then on Friday, we have a full day dedicated to neurotechnology, which is spearheaded by EverythingALS's Indu Navar and many members of her team and her consortium that she's put together. It'll be really another day not to miss. And that will wrap up our 3-day symposium. So thank you so much. This has been such an incredible day 1, and I hope everyone has a wonderful night. Thank you so very much. And thank you, Bob, so much. This has been incredible.
Great day. Thank you.
Thank you.
Thank you, Jen.
Bye-bye.
Bye-bye, everyone.
Bye.
everybody.